Good morning friends, welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe to my channel and also please give valuable comments. You can also visit the playlist. In the playlist I have provided almost all the topics in a very systematic order. I hope this will be very convenient for your continuous study. Now about the hepatitis, we have already completed 3 classes. Today we shall see mechanism, how the virus enter inside the hepatocyte, and in the hepatocytes how intelligently it will make use of the hepatocyte human body cell, hepatocyte cell machinery for its convenience. So that we will going to study today. Hepatitis B virus contains a double-stranded DNA and it requires a reverse transcriptase enzyme to produce the DNA from the RNA template. Now, reverse transcriptase enzyme is that DNA polymerase has contained the reverse transcriptase enzyme quality. So, what do you mean by reverse transcription? Reverse transcription means usually in all the cells under the normal condition, the DNA is used as a template to produce the RNA, isn't it? Only from the DNA the RNA can be produced, and in this process so many mRNAs are produced, and this is called as transcription. We know that. Okay, and that means DNA is used as a template to produce a mRNA. This will be done by the help of the RNA polymerase enzyme. In the normal body cells, the production of this particular mRNA from the DNA template is done by an enzyme called as RNA polymerase enzyme. Okay, and that particular enzyme is called as DNA dependent RNA polymerase enzyme. That enzyme is called as DNA dependent RNA polymerase enzyme. Later, this mRNA, which carries genetic matter, produces the protein corresponding to that particular gene, which is called as a translation, translation. We know that. So protein produced in the ribosomes later. These protein brings the activity of the body for which it is targeted. Okay. This is happening in a normal cell. But in case of reverse transcription, what happens? Reverse transcription is the reaction where from mRNA a double-stranded DNA is produced. See, exactly opposite. From a DNA, there should be production of a mRNA. From the mRNA, there is transport mRNA are produced. Then ribosomal mRNAs are produced. They go to the ribosomes and produce a protein. Okay, but here the ulta takes place, opposite takes place. Reverse transcription is the reaction where from the mRNA, the double-stranded DNA is produced. And this is possible only when in the presence of an enzyme by name reverse transcriptase. Transcription is reversed in the presence of an enzyme called as reverse transcriptase enzyme, which is also called as RNA dependent RNA polymerase. Okay, so reverse transcriptase enzyme, this is also present in HIV virus. In HIV virus also this particular enzyme is present, reverse transcriptase. It is also present in hepatitis B. Now, reverse transcriptase enzyme has got 3 important activities. What is that? RNA dependent DNA synthesis, RNA degradation, and DNA dependent DNA synthesis. It can carry out all these 3 activities. Okay, now hepatitis B virus contains dsDNA, double-stranded DNA. Hepatitis, because immediately after entering inside the hepatocyte cell, the circular DNA is get converted into double-strand DNA. So it now contains double-strand DNA which is present in the coiled matter along with an RNA polymerase enzyme. This dsDNA, double-stranded DNA, is not completely coiled and it is enjoined at the ends. Through the endocytosis, the virus enters inside the hepatocyte. Now, the outer capsid gets dissolved and the central core contains the genetic material that get liberated out and passes into the cytoplasm through the microtubules. That means DNA of the virus now is entering into the cytoplasm of the hepatocyte cell. The core section also get dissolved and only the double-strand DNA along with the RNA polymerase enzyme enter inside the nucleus through the pores of the nuclear membrane into the nucleoplasm. Now it enters further inside. Now the chromatin material relax little bit and start coiling. Chromatin material of the virus. By using the local proteins present in the nucleoplasm there are so many proteins are present. Now this viral DNA, it uses the local proteins. Okay, this is what I am telling. It uses the cell machinery for its convenience. By using the local proteins present in the nucleoplasm of the incompletely connected viral double-stranded DNA gets completed. That means previously that double-stranded DNA was not completed, it was opening at the ends. Now by using that proteins, it is completed. Okay, it now totally circular, completed into circular form. Now this is called as a cccDNA, central closed circuit DNA. Okay, RNA polymerase enzyme present there. Now it produces 4 mRNA by using this DNA. Okay, that particular RNA polymerase, so that produces 4 mRNAs. It can produce 4 mRNAs at a time. Therefore, it is called as polygenomic RNA. The largest mRNA, which is longer than the viral genome, is used to make the new copies of the genome and make the caspid, core protein and the viral DNA polymerase. Okay. Now what happens? mRNA polymerase, RNA polymerase, because that particular, this has got capacity to produce all these things. First it produces RNA polymerase, now it produces DNA polymerase. These 4 viral transcripts undergo additional processing and then go on to form the progeny of the virons that are released from the cell or returned to the nucleus and recycled to produce even more copies. Now everything is under their control, so they now go on producing it. So these pregenomic mRNA, which is the largest mRNA among the 4, now moves out of the nucleoplasm into the cytoplasm and reach the ribosome and the Golgi bodies of the hepatocyte to produce the proteins, whichever it is required according to its convenience, for the genes present in the DNA strands of the virus. As a result, the protein by name DNA polymerase is produced. Now, once this DNA polymerase is produced, everything is under its control. This DNA polymerase enzyme performs 3 functions: priming, reverse transcription, and production of the structural proteins, which is required for the virus to grow. Newly formed protein, which is coding for the viral part, DNA polymerase enters into the nucleoplasm. It starts acting on one mRNA and does the action of priming by replacing a codon, and first DNA strand is produced. Again, by using terminal part of the mRNA, second strand of the DNA will be produced or completed. Likewise, there is production of double strands of DNA by using the mRNA strand. Hence, it is called as reverse transcription reaction. Okay, double strand of the DNA is produced by using mRNA strand. Previously, mRNA is there. Then that mRNA produced DNA polymerase. From the DNA polymerase is now doing production of the double strands of DNA. Therefore, now this is called as reverse transcription reaction. These viral DNA gradually enters into the nucleus, get internalized or integrated with the host cells. In the due course of the time, it completely takes the charge of the host nucleus and conduct as for the wish of the viral DNA, and gradually it destroys the hepatocyte, which may result in chronic viral hepatitis B or cirrhosis or hepatocellular carcinoma. This integrated DNA start producing its own mRNA and start producing the proteins which forms the viral body parts like caspid, and as a result there is formation of the viral molecules. So simple. It uses the body hepatocyte machinery and start producing those proteins which is required for the production of the virus or multiplication of the virus. So now, what are the viral proteins? Now, viral proteins, there are mainly 3 types of the viral proteins and particles are there, which is responsible for the formation of the structure of the virus: double strand, nucleocaspid core, and spherical filaments. Double strand genetic matter, DNA matter, it contains mainly three important antigens by name HBV-S, AG, HBV-C, and HBe-AG. AG stands for antigen. Now this is also called a spherical or core area, therefore these are called as core antigens. From the nucleocapsid area, it contains HBC, AG, and HBe-AG. Filamentous area, this coating the material and contains HBs-AG. Now this is total process how the things will going to take place. If you want to understand, this is the handmade picture or figure, schematic representation, how the things will going to take place. See, first the surface coiled DNA is there, then it is get completed, then it enters inside the hepatocyte, endocytosis takes place. Okay, so then what happens, there is formation of the RNA polymerase enzyme. Once the RNA polymerase enzyme is formed, then there is a DNA structure of the hepatocyte, it will going to enter. From there it forms the DNA polymerase enzyme. So like this, how the things are taking place, it is explained in the schematic pattern. So this is also further the viral DNA and viral RNA, how it forms, what do you mean by priming. Okay, everything is explained here. So in the next class we shall discuss about the clinical manifestations. Thank you very much.